a little bit about what we learned. So in the last class, we started discussing oxidation and reduction reactions. In which type of reaction do we lose electrons in our products? Oxidation or reduction? Oxidation, right. We gain electrons in our product in reduction, right? Okay, so we talked about that. And we talked a little bit about catabolism and anabolism, and then we discussed the process of glycolysis, which is the first process in cellular respiration. We haven't talked about cellular respiration completely yet, but we did talk about glycolysis, which we said can happen both in an aerobic and in an anaerobic environment, right? So regardless of whether or not you have oxygen, the cell, when it's breaking down glucose, will start with glycolysis. Where does glycolysis occur in the cell? Where? The cytoplasm of the cell is where glycolysis occurs. So glucose goes into the cytoplasm, and the first thing that has to happen, we need to use an ATP molecule to change the glucose molecule into glucose 6-phosphate. What's the purpose in this? Why do we have to change it right away?
So we can store it.
So that it can't get out of the cell. You're close. So you can't get out of the cell. You want to trap it inside the cell, so now it's committed to be broken down. Okay, then we add another phosphate from another ATP, and we generate a symmetrical molecule, because after that, we're going to take and break down our 6-carbon molecule into 2 3-carbon molecules. So that second ATP's job, that phosphate, is there to make that molecule that we've created, the 6-carbon molecule, symmetrical, so that when it's cut into half, we can metabolize it separately in each pathway. So through the process of glycolysis, we put in one glucose molecule, and in the end of this metabolic pathway, what do we produce? We produce what? 2 pyruvic acid molecules, right? We also reduce how many NAD molecules do we reduce? 2. So when I'm saying that we reduce them, I'm saying that we start with NAD and we gain electrons to become NADH plus H. So that's reducing NAD. So we get 2 reduced coenzyme carriers. And how many ATP do we produce net? 2, because we see that we produce 4 ATPs here, but we had to put 2 in initially, and that's why we only net 2 ATP per glucose. So the process of glycolysis, if you don't have oxygen readily available and you do this anaerobically, you're not going to produce a whole lot of energy. You're only producing 2 ATP per glucose. But what happens is we have to do something with the NADH plus H molecules that we've created. If we don't have enough oxygen available, we have to do something with the hydrogens there, because we don't want to saturate our coenzyme carriers. They're used for other things, not just for this process. And so therefore, we got to get rid of the hydrogens on there. So here's what happens. We go through lactic acid fermentation in humans. We have 2 pyruvic acid molecules, so this reaction happens twice. The pyruvic acid molecule is going to take on the hydrogens from the NADH plus H. So that means that when the pyruvic acid gains the hydrogens and the electrons, it's reduced to lactic acid. Remember that oxidation and reduction reactions, known as redox reactions, are coupled. So pyruvic acid gains the electrons and hydrogens, NADH plus H loses and becomes NAD again. And now these coenzyme carriers are not saturated and they can help with other processes. Otherwise you would end up basically stopping a lot of processes if you blocked up your NADs. Oops. Okay, what would we produce? What would we produce if we were a yeast cell, not lactic acid, but no, it's a yeast cell. Alcohol, ethanol, right? We don't do that though. We produce lactic acid. So essentially we have, in this process, per glucose, we would have 2 lactic acid molecules produced. We would get 2 oxidized NAD molecules. All right, then we talked a little bit about the process of making and breaking glycogen down. Creating glycogen and breaking it down. So here we have skeletal muscles. We know skeletal muscle cells and liver cells, they both can store glucose as glycogen. So what happens, the glucose goes into a cell, and if that cell needs the energy, it can proceed through the glycolysis pathway. If not, then in liver cells and skeletal muscle cells, we can take our glucose molecule, our glucose 6-phosphate, convert it to glucose 1-phosphate, remove the phosphate, and have it bind to our long glycogen molecule. Just add another glucose to it. This first pathway that we see is the process of glycogenesis, where we're creating glycogen. But when we take the glycogen and we need glucose for some reason, maybe the cell needs energy, or if it's a liver cell, maybe blood sugar levels are too low, so the liver has to release the glucose into the bloodstream, then what we would do is add a phosphate, get glucose 1-phosphate and glucose 6-phosphate finally. This is the process of glycogenolysis, as illustrated by number 2. If it's a liver cell, that glucose 6-phosphate can be converted back to glucose and then released into the bloodstream. The liver has an enzyme that skeletal muscle doesn't, and what is that enzyme? Glucose 6-phosphatase. Glucose 6-phosphatase that removes the phosphate, and now glucose can be released. So glycogenesis and glycogenolysis can really only occur in those 2 places, the liver cell or skeletal muscle cells. And only the liver has glucose 6-phosphatase that can release glucose into the bloodstream. That's pretty much where we left off in the last class. Do you have any questions about those things for now?
The top of that diagram is an example of glucogenesis or glucogenolysis, because I thought you said both.
It is. Which number illustrates glycogenesis, 1 or 2?
1.
1. Number 2 illustrates glycogenolysis, right? You're breaking the glycogen down.
All right, thank you.
Sure. Any other questions? All right, so now we're going to talk about another process, which is pretty interesting, called gluconeogenesis. This literally means that we're making new glucose, we're generating new glucose from something that isn't a carbohydrate. So we have over here a skeletal muscle cell. When that muscle cell is exercising and it needs a quick energy, it's going to rely on its glycogen stores. And the glycogen, we broken down to glucose 6-phosphate, we go through glycolysis, and we get our pyruvic acid molecules produced, which then the pyruvic acid will be reduced to lactic acid. The skeletal muscle cell can't keep that lactic acid though, because that would alter its pH and cause a lot of problems. So the lactic acid is released into the bloodstream and carried back to the liver cells. And in the liver cells, the lactic acid can go backwards through the glycolytic pathway. So if let's see here. So basically in the liver, the lactic acid is going to be converted back to pyruvic acid through an enzyme called lactate dehydrogenase, LDH. This is actually one of your cardiac enzymes too. They look for this in the blood if they think somebody's suffered a heart attack. LDH they look for. But the name lactate dehydrogenase means that we are taking away the hydrogens from the lactic acid to produce pyruvic acid. And of course, where does the hydrogen go? The NAD is going to pick up the hydrogens to form NADH plus H. So now lactic acid, is it oxidized or reduced back to pyruvic acid? It's what? Yeah, see, you have two hydrogens on the lactic acid. Pyruvic acid has lost those electrons in the hydrogen, so we oxidize lactic acid back to pyruvic acid. In the process, we reduce NAD to NADH plus H. Then what happens? We go all the way back. See the dotted blue arrows here that are going the other way? We go all the way backwards through the glycolytic pathway. We produce glucose and ultimately glucose 6-phosphate, and glucose 6-phosphatase can allow the glucose to be freely released back into the bloodstream, and then it travels back to the skeletal muscle cell. And this is called the Cori cycle. It's a cycle because it's a complete circle. We end up with where we started from. So again, skeletal muscle cells, they exercise, they produce pyruvic acid, there's not enough oxygen available, they produce lactic acid, which is released in the blood, picked up by the liver cells, and then lactic acid will be oxidized back to pyruvic acid, and then pyruvic acid ultimately will be converted back to glucose 6-phosphate. Glucose 6-phosphatase releases that phosphate, and glucose is released into the bloodstream and goes back to the skeletal muscle. So that's how we produce new glucose from something that wasn't glucose. It was lactic acid, but now it's glucose. So it's usable. It's like a recycling kind of thing in a way. Do you have any questions about that?
What were you saying about the lactic acid coming from the bloodstream to
Lactic acid goes from the blood to the liver cell, and then the lactic acid is oxidized back to pyruvic acid, and then pyruvic acid is converted back to glucose 6-phosphate. And then the enzyme takes away the phosphate, so free glucose can be released into the blood and goes back to the skeletal muscle.
This cycle only initiates under certain conditions, so like digestive conditions.
We're talking about muscles. So this is where if you're exercising and if you're not, like if your skeletal muscles aren't conditioned enough aerobically and they can't extract enough oxygen to go through cellular respiration to make an adequate amount of ATP, you end up producing the lactic acid through the fermentation pathway. That's the situation. When you're tiring out your muscles, they don't have enough oxygen available and they're generating lactic acid, they're building it up. Then that has to go somewhere to be processed, and it goes to the liver, which converts it all back to glucose. Any other questions? Okay, so this will come in handy when we talk about the skeletal, the muscular system, because the muscles do utilize, certain fibers utilize oxygen better than other fibers do, and some of them are more efficient metabolically, and you can train these fibers to be more efficient and so forth. So this will come in handy. Understanding these concepts will help you with the muscular system in the future. So now, we've talked about what happens if we don't have enough oxygen available. We go through glycolysis, we We go through the fermentation pathway. We know it's not super efficient, and we produce lactic acid, and you can't really sustain activity that way. Remember we said when oxygen levels, for example, fall in cardiac muscle tissue because there's an occlusion or something, atherosclerosis, then you get pain, which is from the lactic acid that's being generated because the cells can't make their energy efficiently, and they're producing lactic acid. Now, the remainder of this is all about what happens if we have enough oxygen available. And this begins basically the process of cellular respiration, where cells take in oxygen and they make carbon dioxide and get rid of it. So basically, what happens to pyruvic acid if we have enough oxygen available? If pyruvic acid is in the presence of oxygen, then it is oxidized to acetic acid. Does anybody know what acetic acid is? Probably have it in your cupboard at home. What is it? No, that's acetone. But acetic acid is vinegar. And we produce vinegar. We actually produce vinegar in our bodies as a byproduct of this reaction. So pyruvic acid is converted to acetic acid, and the acetic acid combines with another coenzyme carrier called coenzyme A to form acetyl coenzyme A. Now, in the process, again, this is oxidation reduction, we're going to be producing NADH plus H molecules. So I'm going to ask you, what is oxidized here? Is it going to be the pyruvic acid or the NAD? We start with, in pyruvic acid, we start with 3 carbons. When we get acetic acid, we only have 2 carbons. So when we start here, we start with NAD, we end up with NADH plus H. Which is oxidized, the pyruvic acid or the NAD?
Pyruvic?
Right, pyruvic acid, because it loses the carbon, it loses electrons, right? So it goes from 3 carbons here to 2 carbons here. So pyruvic acid is oxidized to acetic acid. NAD is reduced to NADH plus H. Now the trick, how many acetyl coenzyme A molecules do we produce per glucose? 2. Yes, I told you pretty much you stay with that number for most of this. How many NADH plus H molecules do we produce? 2. Very good. Why? Because remember, per glucose, how many pyruvic acid molecules do we produce in glycolysis? 2. Exactly. This reaction happens 2 times per glucose. Okay, so now acetyl coenzyme A. Coenzyme A is basically going to carry our acetic acid into the mitochondria, which we know is the powerhouse of the cell where most of our ATP is produced. And in the matrix, which is very much like the cytoplasm inside of the cell, the matrix is inside the mitochondria. In the matrix, we have something called the Krebs cycle or citric acid cycle. Citric acid is found in things like lemons, limes, and oranges. It's the same stuff. But it's called that because the first product that we produce is citric acid. But Krebs was the man who named it, so you can use those terms interchangeably. They're the same process. So essentially what happens, we have 2 carbon acetic acid molecule. Coenzyme A leaves, goes back to pick up more acetic acid. It's a carrier, right? It's a coenzyme. So we have 2 carbons, we have 4 carbon oxaloacetic acid that combines with it. Now we have a 6 carbon molecule of citric acid that's produced. And then there's a series of other reactions because it's a metabolic pathway. And ultimately we end up with what we started with, which is why it's a cycle. So now don't be worried about this. You don't have to know every intermediary. Again, I'm going to write with, oh, you know what? I got all those papers in order too. Good thing it's not a big class. So basically, the citric acid cycle or the Krebs cycle, there's only a couple of components that you really need to know that I'm going to point out to you so that you can tweeze out what's important. How many times will this cycle occur per glucose molecule? Twice. Because, remember, we produce 2 pyruvic acids, we produce 2 acetyl coenzyme A's, and we're going to have to go through this cycle 2 times. So everything that you see on here, we're going to double. All right, so in the process of the Krebs cycle, when all is said and done, okay, how many ATP molecules have we produced? 2. You see 1 here, and then of course the cycle will happen twice, so there are 2 ATP. How many NAD molecules are reduced to NADH plus H? 6, right? Yeah, you got 1, 2, 3, multiplied by 2, that's 6. So that was the only place that we got tripped up a little bit there, right? That's the only one that breaks the 2 rule. Because FAD, which is another coenzyme, and this is the only place we see it, FAD becomes FADH2, it reduces. And how many of those do we produce in the Krebs cycle for glucose? 2, just 2. So you see it 1 here, and then you'll have another one the second turn of the cycle. So that's pretty much the Krebs cycle in a nutshell. So if we put the ins and outs of the Krebs cycle, and then we'll talk about it on your handout. So if you wanted to get your handout out again that I gave you guys on Tuesday, it would be good. Because we're going to go over that next. Okay, so per glucose, okay, we have 2 molecules of acetic acid. And, you know, really, the only reason that I'm putting this on here is because I just want to remind you that the first product produced is citric acid. But, of course, really, I guess our end product would be oxaloacetic acid, but I'm putting this. We have 2 citric acid molecules produced. We have 2 ADP and phosphate. We get 2 ATP produced. We had 6 NAD molecules going in and 6 NADH plus H coming out. And we had 2 FAD molecules going in and 2 FADH2 molecules coming out. So this was in, out. So now if we take a look at number 2 and number 3 on the handout that I gave you guys. Number 2 talks about the transitional step where pyruvic acid is converted to basically to acetyl coenzyme A. So 2 3-carbon molecules of pyruvate are decarboxylated, means that we take away a carbon, right? We go from 3 carbons to 2 carbons in acetic acid, and broken down into 2 2-carbon molecules of what? I just answered it. Acetic acid, right? And 2 molecules of acetyl, so that combines with coenzyme A. 2 molecules of acetyl coenzyme A enter the citric acid cycle, and then in doing this, as we said before, 2 molecules of NADH plus H are produced. So, we go back.
Do you have an extra paper?
Oh. Should. made one for everybody, so I had to have an extra. So again, if we just wanted to simplify this process in terms of what goes in and what goes out, this is our transitional step. So what goes in for this one, we have 2 pyruvic acids. We get 2 acetic acid molecules plus 2 coenzyme A's that are going to be carrying the, and we combine the name and call it acetyl coenzyme A. And we have 2 NAD molecules that are reduced to 2 NADH plus H molecules. And again, pyruvic acid here, this is the pyruvate oxidation step. It's oxidized. And NAD is reduced to NADH plus H. So there's the redox part of this. Okay, so next we have the Krebs cycle part. So in the citric acid cycle, each of the 2 acetyl coenzyme A molecules are converted into 2 molecules of what? First thing that's produced?
Citric acid.
Citric acid, right. The first product that we produce. Acetyl coenzyme A becomes citric acid when it combines with oxaloacetic acid. The citric acid cycle occurs in the mitochondrial what? Do you remember what I told you? Matrix, yes, the matrix, which is really a lot like the cytoplasm inside a cell, but it's specifically the gelatinous inner part of mitochondria. Each molecule of, and you know what? Let's change this around. Let's put cross out each molecule and just put per glucose, comma. So cross out each molecule of, and just right above it put per glucose, comma. Acetyl coenzyme A yields how many ATP? Per glucose. That means that the cycle has to occur twice per glucose. So we have 2 ATP, how many NADH plus H? 6. And how many FADH2? 2. That is correct. Good. Alrighty. So any questions about that? I think really between this stuff that I wrote on the board and then this kind of narrative study guide, this is the main thing that you need to know about these processes. So it makes it a little bit more condensed and it doesn't look so overwhelming because these diagrams just really look intimidating. There's so much written on there because they draw out the structure of everything. What we really care about is what's coming out of this. What are we getting from this cycle? What are we producing? That's the key thing. So we are going to now enter into the last process of cellular respiration and the process by which we make the largest amount of ATP. Because right now, how many ATP molecules have we made through all those reactions? How many in glycolysis? Any in pyruvate oxidation?
2.
We made none there, but in Krebs cycle we made 2. So we only made 4 ATP. All that talking, all those diagrams, all those reactions, only 4. So electron transport chain, it's true, right? I mean, I always think about that. Electron transport chain is the last process, and it generates the most ATP. But how? Well, let's think about it. What have we produced the most of in the reactions that we've done so far? What have we made the most of? We haven't made much ATP, but what? Yeah, NADH plus H. Let's count up how many we've made. How many did we make in glycolysis? 2. 2. In glycolysis, how many in pyruvate oxidation? 2. And then how many in Krebs?
6.
For a total of 10. Yes. And we've made how many FADH2 molecules? 2. So we've got a total of 12 coenzyme carriers that have hydrogens that they're carrying around. So there must be some reason and importance for this. And this is where it comes in handy. So first of all, the electron transport chain is found in the inner membrane of the mitochondria. Remember, the mitochondria has an outer membrane and an inner membrane. The electron transport chain, and there are hundreds of them. This is just showing one, but there are hundreds of them in one mitochondria. It consists of 3 protein pumps, the first, second, and third. It doesn't have a special name. And what happens is, down here in the matrix of the mitochondria, the NADH plus H molecules and FADH2, they're going to dump off all their hydrogens. So they're going to become oxidized back to NAD and FAD. So I tell people this colorfully each semester, but it's like a love story. You know, it's like you have to go off and do your duty somewhere, and you may be separated from that who you're most connected to, but then everybody does their job, and then God willing, we come back together in the end, create something beautiful. And that's what's happening here. We have the oxidized NAD and FAD molecules. They dumped off their hydrogens. They're going back to pick up more hydrogen in the future. Now, hydrogen itself, we know, has an electron with it. And what happens is here, in this inner membrane of the mitochondria, also known as the cristae. Cristae is another name for that. The hydrogen and the electron are separated from one another. And the electron is going to be passed from the first to the second to the third protein pump. So what happens here is when the electron is associated with the first pump, that first pump gains an electron. So it is reduced. Then when it loses the electron to the second pump, the first pump is what? Oxidized, the second pump is reduced. Very good. Then the second pump loses its electron, becomes oxidized, and reduces then the third pump when the electron is passed. Now, what's the purpose of this? Well, basically, if you recall, anytime you have reactions taking place, be they oxidation, reduction, endergonic, exergonic, whichever, you're always, any type of reaction always generates some type of energy. It could be heat energy, it could be, some type of energy. And so that's what's happening here. When we pass the electron from pump to pump to pump, it's generating energy. And we need the energy because that energy is going to make it so that we can take the hydrogen, which notice it has a plus sign on it now because it has lost its electron, so it's positive by 1. Okay, so the hydrogen that is going to be pumped into what we call the intermembrane space between the outer and inner membrane against the gradient. We want to keep a real high concentration of hydrogen in the intermembrane space. Okay, so that's what the electrons being passed from pump to pump to pump does. Through a series of redox reactions, we release energy needed to pump the hydrogen across the membrane into the intermembrane space. Then what happens, the electron has to be picked up by something once it's done with the last pump. It has to be picked up by something. Otherwise, this electron transport chain would stop. It would back up and it would stop, and then it wouldn't be functional. And this is where oxygen comes in. Oxygen is what we call our final electron acceptor. When the electron is released from the third pump, oxygen is going to be there to catch it. We know oxygen's greedy with electrons. We saw it in water too, right? is taking on more of the electron from the hydrogen and polar covalent bonding. Remember? Oxygen's greedy. So it takes on the electrons, and if we don't have enough oxygen, that pump would stop, it would back up. That's why we need oxygen for this process to take place. Because without it, you would not be able to keep the pump going. Alright? So now what happens? Hydrogen in high concentration in this intermembrane space can only pass through one channel called the ATP synthase. And it is actually an enzyme. But it's the only way that hydrogen can get from the intermembrane space back into the matrix again. So it rushes through there because concentration's so high, it really wants to, go from high concentration to low. Rushes through there, and in doing so, that provides energy to make ATP from ADP and phosphate. So electron has its job to provide the energy to move the hydrogen into the intermembrane space so that hydrogen can do its job, rushing through the ATP synthase to make ATP. And now, the oxygen having collected 2 electrons, okay, each oxygen collects 2 electrons from the third pump, has 2 electrons, the hydrogen wants its electron back. So, it attaches to the oxygen and you make molecules of water. But they're finally reunited, but something new is created. I know, it sounded more exciting when I first started than what it actually was. I'll explain that for you again one more time, and then we can look at what goes in and what goes out. Okay, let's see here. So, electron transport chain, we're going to see consisting of 3 protein pumps in the cristae, or the inner mitochondrial membrane. Hydrogen is dumped off by NADH plus H and FADH2. So those coenzymes are oxidized, they lose their hydrogens and lose their electrons. Hydrogen separated from its electron. The hydrogen's electrons are passed from pump to pump to pump through a series of oxidation reduction reactions, which then provides the energy to pump hydrogen against the gradient into the intermembrane space. When the electron gets to the third pump, it is accepted by a molecule of oxygen, which is the final electron acceptor. Then hydrogen is able to flow with its gradient through the ATP synthase back into the matrix. In doing so, it generates ATP, provides the energy to make ATP. The hydrogen then combines with the oxygen, which has its electrons, to form a molecule of water. And that's basically the process. Now, through this process, and there is a diagram I'm not going to hold you responsible for, but if you're interested in like how you generate the ATP from how many hydrogens and so forth, you can look at that. But I'm not going to hold you responsible for it, but I included it in the notes just in case you were curious. So for this last process, we have to make a different label here. Now this is actually called, so this is the electron transport part. Okay, that itself doesn't provide, it doesn't make any ATP, electron transport. But this process by which our hydrogen goes through the ATP synthase, that process is called chemiosmosis, and that is where you make your ATP from. So it's not really electron transport, but it's chemiosmosis that makes the ATP. Okay, so what goes in? Well, we started with 10 NADH plus H, and now we've oxidized to 10 NAD molecules so they can go back and pick up more hydrogen. We had 2 FADH2 molecules, which have been oxidized to FAD. And then we have big number, 32 to 34 ADP and phosphate yield 32 to 34 ATP. So we have a lot of ATP that can be produced through this process, definitely. So in total, if we produce 32 to 34 ATP in this process, how many we produce in glycolysis again? How many ATP in glycolysis? 2. How many in Krebs? 2. So that's a total of 4 additional ones. So we have the potential per glucose molecule to make about 36 to 38 ATPs. So if we look at the last paragraph, point number 4, the electron transport chain on your handout, this repeats what we just talked about. The reduced electron carriers NADH plus H and FADH2 are what? Reduced or oxidized? Oxidized, and give their electrons and their hydrogens, right, because that's attached to the electron transport chain. The electron transport chain is located where? Where do you see it? Either the inner membrane or the cristae, right? CRISTAE, CRISTAE, cristae. Electrons travel down the chain. 1, 2, 3. Released energy transports what across the membrane into the intermembrane space? Huh? Yeah, hydrogen. And at the end of the chain, electrons are accepted by a molecule of what? Oxygen, final electron acceptor, right? Which combines with the hydrogen to make a molecule of H2O, water, right? To make a molecule of water. Finally, ATP is produced as what returns back into the matrix or comes back across the membrane through the channels of the ATP synthase. Hydrogen again, right? Does that make sense? Anyone have any questions?
The last link was hydrogen.
Yes. Yeah, so like I said, I stopped making this a take-home quiz for General Biology 101 because there were just too many tiers. And when you have a class of 100 people and a small office, there's just not enough room for everybody to come in and be like, I don't understand. I don't know. I'm going to fail it. I'm like, it's open notes. I don't know where my notes are. Oh, that was good. I remember that. That was like my first year that I taught. And I was just like, oh Lord, this is not worth the headache. Okay, so total ATP from one sugar molecule. Again, how many ATP did we produce in glycolysis? How many in Krebs? 2. Electron transport and chemiosmosis, specifically the chemiosmosis part is where we produce. We have about 32 to 34, right? For a total of how many?
36 to 38.
36 to 38, right. That's your total. If you add all those up. Whew. thank goodness the rest of this isn't so bad. All right, so let's just finish the last couple things before we go into the lipid part of things. We're gonna take a break. So before that, in all the processes that we talked about generation of ATP. There are two ways in which we've made ATP here. Either through the process of what we call direct or substrate level phosphorylation. This is how you make ATP in glycolysis and in the Krebs cycle. So why do we call it substrate level phosphorylation? If I go back, all the way back, I'll give you an example here. In glycolysis, okay, notice where we're making ATP here, and here we're making it the same way. Now notice that we have 1,3-biphosphoglyceric acid. The 1 and the 3, if you remember, just indicates that we have a phosphate on the first and the third carbon, right? So we have two phosphates here, hence the name biphosphoglyceric acid, because we have two phosphates. Notice that, okay, this is the substrate of this reaction. We end up with 3-phosphoglyceric acid, which means what's happened? We lost the phosphate, right? Where did the phosphate go? Well, the phosphate was given to ADP to make ATP. And so that's why we call it substrate level phosphorylation, because we're taking the phosphate off our substrate to make our ATP. That's why we call it that. And this happens in glycolysis and it happens in Krebs. Got it? You have any questions? Okay, so then the other way that we can make energy is, and this is how we do it in electron transport and chemiosmosis, is through oxidative phosphorylation. So this type of making of ATP, we call it oxidative because we're talking about the fact that we're using a series of redox reactions as we pass the electron from pump to pump to pump to generate the energy to get the hydrogen here into the intermembrane space so that we can ultimately generate the ATP as it passes through the synthase. So the idea is that we're using redox to set up the situation to ultimately make the ATP. And that's why we call it oxidative phosphorylation, not substrate level. We're not getting phosphates from any type of substrate here. We're getting it because we've set up a situation where we have a high concentration of hydrogen, and then that moving of the hydrogen is going to ultimately make the ATP. Do you have any questions about that? There's really only one last point that I want to bring up here, and then the rest of this, as I said, I'm not going to really hold you responsible for it, but basically, this particular part right here is going through some of the accounting of like how many hydrogens generate ATP and so forth and so on. So everything that's on this sheet in terms of numbers of molecules of things that are produced, ATP and so forth, that's what I'm going to ask you for an exam. But I could have a bonus question about this. In other words, I'll have a bonus question about this. What we've described is what most classes are going to teach you in terms of how many ATP you get from a glucose molecule. But truthfully, that's under laboratory conditions, which is where they learned about this process in the first place. Remember that the ATP, much of it is made within the mitochondria, and some of that has to be transported out, which requires energy. And so we don't net quite that many that we've talked about here. In fact, what we produce with electron transport and chemiosmosis is really only about 26, not 32 to 34, but 26 ATP. And then through glycolysis, we still have 2, and through Krebs, we still have 2. So we would have probably per glucose molecule, under normal physiological conditions, about 30 ATP that are generated per glucose. But again, that would be a bonus question that I would ask you about. These numbers that I gave you on this is what would be in the exam. Okay, but a bonus question would be, you know, how many, you actually do produce less ATP in electron transport and chemiosmosis than what we talked about here. That make sense? No questions about that? Yeah, so in truth, you're gonna have, under normal conditions, what you guys are probably making in your cells now, 26 ATP from chemiosmosis, 2 from glycolysis, and 2 from Krebs cycle for a total of 30. All right, and then again, this table I'm not going to hold you accountable for, but if you're interested to find out, like, okay, how many coenzymes, how many hydrogens generate 1 ATP, then you can kind of look at the calculations. Some people like to see that, but I'm not going to hold you accountable for that. All right, go ahead and take 10 minutes. You deserve it, and I think you're following us.